Okay, so I uploaded this new assignment to your team. So let me kind of bring that up. It's right here. And then if you look at the assignment, it's basically your three assignments combined into one. So it's right here. And if you look at the student view, where did I upload it? Oh, no way. Did I not upload the assignment? Oh, I haven't uploaded it yet. That's why. All right. So again, this is what we're going to use. And let's check it out. Once it says uploaded, you're going to see it's right here. Okay, so basically this assignment is your three assignments combined into one. I took some questions out, so questions related to the workbook, like the big picture questions, some questions where you have to like fill in like which is heterozygous and genotype. I took everything out, so it's all free response questions. So like the first one, if you have started on these questions, that's fine. You can totally just copy and paste whatever you have into this. You can either edit this document and put your responses into this document, or you can just do this from scratch and just upload your own version into here. So this assignment basically encompasses everything from the beginning of the genetics unit to dihybrid crosses. So like the beginning, this is from like your first assignment, what process allows the diversity of genetic material, makeup of sex cells, and then this all goes down into talking about monohybrid and dihybrid crosses with laws of segregation, laws of independent assortment. Okay, so that's your assignment. I set this due date to I think next April 15th. So next week, I think Wednesday or something like that, or next Tuesday. Let me see. Next Wednesday. So you have a week to complete this. Yeah, that's about it. The pedigree assignment, I can bump that one up a little bit to be due a little bit later too, and that's okay. I know it says 14th. Let's just kind of change this right now and make this due not next Tuesday. Let's make it due like that Monday on the April 20th. There we go. So everything's good, and I think by then we'll pretty much wrap up this unit pretty quickly, and that's about it. So if you have any questions, I don't know who's watching this stream right now. I know there's like 5 people here, but like just let me know, or if you can just let me know who is here, and that'd be great as well. Yeah. All right. Dihybrid crosses. Boom. Dihybrid crosses, if you watched the video already, is really... Hey Joel. Hey Haley. Dihybrid crosses are just really you have 2 different alleles, and you look in the combination of those. So if you were to think about our chromosomes, right? Chromosomes have tons of DNA, and within the DNA, the chromosomes, there's tons of genes that's embedded. So when we just look at that Mendelian trait of just like your widow's peak or your earlobes attached, or that's just that one allele. So it's really easy to separate. Hey, Hannah. So that's really easy to separate. But as you look at the reality of your chromosomes, there's tons of genes in all of our chromosomes, right? There's 46 chromosomes in us. Every chromosome has a lot of genes attributed to different things. So if you think about the mixing of all these during meiosis, right? The crossing over, the mixing of all these, that's a crazy combination, a crazy amount of combination that you can have. And that's really what's, what's really happening. That's why if you were to look at you and your siblings, right? You and your siblings don't look exactly alike unless you're twins, but like the amount of sort of diversity that's inside is immense, right? Obviously, if you were to look at like 2 parents that sort of share similar traits, like if both of them have brown hair, or well, if they both of them have like blonde and blue eyes, right, like very recessive traits or whatnot, right, then the offspring are most likely going to be like that. as well, right? If they're sort of similar in physical size, right? That's very similar. If you look at like Yao Ming, for example, for like basketball player, like he's what 7'5", 7'6", or something like that. If you look at his parents, like they're both athletes, right? So like they're all like those genes have to come from somewhere. And also, if you were to look at like Alexander Ovechkin, I don't know if you know, like a hockey player, right? He's a pretty big guy, but if you look at his parents, both of them were athletes and big people as well, right? So that's where it gets passed down. So when we look at dihybrid crosses, now we're not just looking at that one particular trait, but we're looking at the two particular traits that gets mixed. Of course, this is just dihybrid cross. You can have like trihybrid crosses. Like you can have more of these. It's just your Punnett score gets massive, right? Because every trait that you add in, your Punnett score gets bigger because there's more combination. Right, so if we were to look at, let me see if my pen is working. Oh, I think it is working. If we were to look at dihybrid crosses, right, here's an example. So how we set this up is that now if you look at the question, there's two different alleles that's present, so two different letters. And I think the practice here is important for you to get used to how to set this up. Because like when you watch me do it, it's easy, but then when you see the letters, it's like, well, how does all the combination go? So it's really important for you to practice this. So if you look at the first question, in humans, assume spotted skin is capitalized S is dominant over the non-spotted skin, which is the lowercase s, and the woolly hair is capitalized W over the non-woolly hair, which is lowercase w. Cross and marriage between a heterozygous spotted and non-woolly man. So you know that there's two traits here. There's non-woolly and heterozygous spotted with a woolly haired non-spotted woman. So let's just kind of do the genotypes for both of these to start, right? So the male has a heterozygous spotted. So heterozygous spotted is going to be an uppercase S and a lowercase s. And then non-woolly man. So we know that non-woolly has to be homozygous recessive. So this is going to be two lowercase w's. And this is going to be crossing with the female, whose genotype is going to be woolly hair. So what's woolly hair here? With a woolly hair, non-spotted woman. So let me see. Non-spotted woman is going to be two lowercase s's because these are homozygous recessive. Are there any clues about the woolly hair? Is it a heterozygous or homozygous dominant here? Let me just kind of reread the question. Dominant over non-woolly hair. Okay, cross marriage between a heterozygous spotted non-woolly man with a woolly hair non-spotted woman. This is still working. Is it back on? Totally lost internet for a moment here. Well, at least I know you're here, so that's good. Are we good? Is it working? It says it's working right now. I think it just says poor connection. Internet has been so bad this morning. Even like at our staff meeting, people are like in and out. So hey, Flavio, good to see you. Okay, so just resuming this, the quality might not be the best right now, but here's the thing. Let me just write out the phenotypes here, just so I don't get confused here. So this is spotted, and then two lowercase w's. Oh, that's bad. Two lowercase w is non-woolly hair, and then yeah, at least you can just type your question away. Two lowercase s's is non-spotted. And then uppercase W is woolly hair. So when you set this up, this is what you're going to do. You have to look at the combination of what is going to be found in your sex cells, right? If this is your male, this is going to go into your sperm cells, right? So if I were to look at what the law of independence assortment is, what that really means is that each of these, let me change a different color. each of these are going to be separated independently from each other. That's what independent assortment is. That's why it's called independent assortment, right? So that means that if you were to say, if you were to have like a jar that's covered, and inside this jar you have an uppercase S and lowercase s, and you're just reaching in and then you're randomly selecting. What you're going to get is that you're going to either get an uppercase S or you're going to get a lowercase s, right? That's your 50-50. Likewise with this one, if you're going to have a jar of just two lowercase w's, right? So then if you were to reach in and select it, well, you can only get lowercase w, right? Because that's the only combination that you have. So your combination of your traits, how you're going to set this up is that you're going to have one that's going to have an uppercase s and lowercase w, and then you're going to have one like right here, and then you're going to have one that is lowercase s and lowercase w. So that's your two combinations that you're going to have for your Punnett square. So if I'm going to set this up as my Punnett square right here, good thing it's still on the screen, my male is going to have this combination, and I'm going to have this combination. Okay, I'm going to put up my Punnett square, and then for my female, I'm going to look at my combination here. So if you were to put all the traits into a jar again, well, I have two lowercase s's, and you're going to reach in, you're going to select one, and you're going to notice that, well, the only one that you can get is lowercase s, and then you're going to have a jar that is two uppercase w's, and then again, you're going to reach in, you're going to grab one, and you notice that the only thing that you can get is the uppercase w, right? That's the only possibility. So instead of being redundant and having like, oh, right, lowercase s and uppercase w again, you're just gonna do one of them, like this. So your Punnett square for this is actually a lot smaller because these are the only possibilities that you have. So once you have set up your Punnett square, then you just complete your Punnett square, as in find the combination between these two, and then that's your first combination. And then second combination is this one, and then the big W and lowercase w, like that. Okay, so this particular example is you only have two different possibilities. So if you look at what uppercase S is spotted, and then uppercase W is woolly. So this is one possibility, 50-50. Lowercase s is non-spotted, and then uppercase W is woolly. So your offspring is 50% this and 50% this, 50% and 50%. All right. Anise, you had a question. If you're a carrier of the recessive gene like blue eyes, but you don't have the trait, wouldn't it be a higher chance of the offspring? When you don't have the trait, would it have a higher chance for the offspring to have it? Well, yeah, it depends on what you have, right? So, if your spouse is homozygous recessive for blue eyes, then yes, there's going to be a higher chance. If your spouse is homozygous dominant, then there's zero chance, right? Because that dominant trait will just sort of overshadow that recessive trait of blue eyes. Okay, so this is what dihybrid cross is. Let me know. Continue asking me questions in the chat, and I'll answer them once I'm done this. So this is just one possibility, right? Because if you were to go back to the question, we didn't know if the woman was homozygous or heterozygous. So let's do one that's heterozygous. So I'm just going to use a space. I'm going to cross out number 2. I'm going to use the same two traits. So here's my male. And I'm going to cross this. Now, this is going to be a woolly hair. So the woolly hair can be heterozygous. So right now, this was homozygous dominant. This, I'm going to use it as a heterozygous. Okay, so if you look at this again, how would we set up the different traits that we'll have? So that this one is going to stay the same. We're going to have uppercase S and lowercase w, or lowercase s and lowercase w, right? That's what we did. For this one, you can see what you're going to have. The only possibilities that you have is that you're going to have a lowercase s and uppercase w. So we're going to have this and this, or you're going to have the lowercase s, this, and that. right? So those are your two possibilities, and that changes up your Punnett square size, because if you were to put your males, which is here, and here's my female, so let's do that. So I'm gonna set it up here, SW, and my homozygous recessive right here. And then for this one, I'm gonna have my lowercase, uppercase, lowercase, lowercase. So far so good. Complete my Punnett square like this. And I'm gonna have uppercase, lowercase, that's from this one, and then uppercase, lowercase, right? This one and this one. Oh, I shouldn't do that, because then now you're gonna get confused by this, right? Let me just kind of erase that little line underneath. Oh, same for that one too. So then that's that. And then the next two is just gonna be two lowercase, uppercase, lowercase w. And then this one's gonna uppercase, lowercase, 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 lowercase. And then two lowercase w's, or two lowercase s's, I mean, and that's your combination. So once you have established this, then you're going to go back and see, like, what's the phenotype, what's the physical feature of this, what's the physical feature of this, and then that's one, and then what's the physical feature of this and that, and you're going to go through all of this and then look at the percentage of each of these. Okay, that's your dihybrid cross. We actually haven't even gone through the big box yet, which I'll show you right now. But if you have any questions, again, let me know in the text box, and then I'll give you like a couple seconds to just kind of copy this down if you need to. Okay. If I were to go to a blank one here, so you're going to encounter different ones that's going to be two heterozygous. So let's just kind of use the same letters as above, but instead of having homozygous, dominant, or recessive, you're going to have a heterozygous crossing with a heterozygous parent as well. So when you have two heterozygous, your Punnett square gets a lot bigger. So again, how would we set this up to look at all the combinations? Again, if this helps you, imagine that you have a jar, and inside the jar are your traits or your alleles, right? So inside this one are your S traits, right? So if you were to just randomly go in the box or go into the jar and pick one, you either get this or this, right? Those are your two possibilities, right? And then you're gonna go into this jar, and then you're gonna do the same thing with the W's. So you either get an uppercase W or lowercase w. However, you can also have different ones, different combinations as well. So what that means is that you can also have an uppercase S for this combination with a lowercase w that's drawn from this one, right? Because you can have both of them. Or you can have a lowercase s or an uppercase w. Okay, so if you're just drawing these ones from the jar, you can have both of these combinations. So if you look at this and how we would set this up in a Punnett square, you're going to put all of these ones on the side. Okay, and then...
I took a picture of you.
Okay. And then for this one, this is also dihybrid heterozygous, so you're going to have the same combination. So you're going to have an S, uppercase S, and an uppercase W, right? That one to that one. And then you're going to have the uppercase S to a lowercase w. And then the next combination, you have this lowercase s with a big W. And then the last one is the lowercase s with the lowercase w right here. So when you put all of this up into your Punnett square, you're going to see that you're going to have 16 boxes right here. Let me see if I can actually make this nice here. 16 boxes. So we're going to go and create this grid. And then once you have created this grid, then you're going to go through all of your possibilities, right? So you're going to go with this one and this one to fill in the first box. So your uppercase S, and then two uppercase W's. Okay. And then go down this same thing. So uppercase S with a big W, lowercase w. That's why it's very important for you to underline the lowercase letters. Uppercase, lowercase, uppercase, lowercase. Try to write your uppercase first. Uppercase, lowercase, uppercase, uppercase. And then once you fill all of this in, I know it's tedious, but it is really, this is time consuming, but this will give you a really good ratio here. So if you look at this, S, lowercase, here, uppercase, lowercase, uppercase, uppercase, uppercase. And then once you have done this, again, this is incredibly tedious here, right? This should be underlined. And then you're going to get, let me see. Boop, boop, boop. It's gonna, I'm getting lost in these pink letters here. So 2, 2, and then, yeah, that's it. Okay, so once you have completed this, and if you go through your phenotypes, you're gonna see that you're gonna have this 9-3-3-3-1 ratio. So what does that mean is that your 9 is actually going to be your dominant traits. I just running out of space to identify all of these. But if you were to look at all of these, 9 of them are going to be your dominant ones. So like dominant as in like this is uppercase S, uppercase W. So this is your dominant one, dominant one, dominant one, dominant one, dominant, 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 and then that's it. So that should be 1, 2, 3, 4, 5, 6, 7, 8, 9. Perfect, right? 9. The next one is going to be your 3 is going to be your sort of your dominant and recessive. So like uppercase S and lowercase w, uppercase S and lowercase w, uppercase S and lowercase w. That's your 3. And then your next one is going to be the other set, which is your recessive and dominant, recessive and dominant, and then recessive and dominant. So that's your 3. And then your last one, your 1, is your 2 recessive traits. So right here. So then the math part can come into play. Is like, well, what's the percentage, right? So the percentage is here is just really 9 divided by 16, and then 3 divided by 16, 3 divided by 16, and then 1 divided by 16. And then once you have done all this, then you can multiply by 100 to get your percentage. So that's your dihybrid cross. The biggest Punnett square you'll see is when you have 2 heterozygous dihybrid crosses crossing each other. So really, that's all I wanted to talk about today. If you have any questions right now, just let me know in your chat box right now. Hopefully this has helped you. It's always nice to, I know like I have a video on this, but it's nice to just kind of sit through a lesson and just kind of connect with each other and see me in my house with kids running around, so that you know that I'm not just gonna sitting there and do nothing. So let me know if you have any questions or any comments. All right. You have a couple of assignments. We'll do a really short check-in tomorrow. When we answer these questions, is it okay to express? It depends. Yeah, it depends on the question. Okay, so like some questions will ask you for the percentage. Some of them will just ask you for the ratio. So if the question is asking you for the ratio, this is your ratio. The 9:3:3:1. That's your ratio. If it's asking you for the percentage, then you will need to change it into percentage. Okay. Hey Owen, good morning. So you can see everybody's here. This is my morning, right? Yeah, that's right, Charlotte. You're so excited. Any other questions? Owen, I posted a new assignment. If you want to check that out, is basically your 3 assignments combined into one. But I took out some of the questions that are like word problems. Yeah. All right. Okay. So if there's nothing else, I think I gotta let you guys go. We'll have a short stream tomorrow because I don't think there's any content. We'll just do a little check-in, or if there's any questions you want me to go over, we can totally do that. But that's about it. I think I gotta go. Okay, bye. You gonna say bye, Charlotte? You gonna wave? There you go.